Hello, in this video we will learn about the light transduction pathway. So what is the light transduction pathway? And we will also learn about the biosynthesis of the during the Walds visual cycle. So basically we will learn about the Walds visual cycle also. So let's begin to understand without wasting any time. So here is the black body, black object. It means the black color means all wavelength of light which that is come from the sun, which that is the VIBG, or we can understand the violet, indigo, blue, green, yellow, orange and red. All wavelength will be absorbed by this body to show the black color, while the white color means that all radiation will be transmit, reflect from the object, so it means the white color. While the if one only red color will be reflect, so it means the color will show the red. So that means this reflected color, reflected wavelength will go into the lens of the eye and go into the retina for detection of light or detection of some shape or something. It means the angle of the light reflection will affect the retina. So here is the wavelength is shown in the lambda, which that is contain different wavelength, contain different colors. So here is the introduction. Let's begin to understand first of all eye. Eye is just like a camera. So in this a pinhole camera look like structure, because the image will form into the back side of the eyes, posterior side. So here is the cornea, which that is a transparent, used for the light entrance. While here the pupil. Pupil is used for the dilation and constriction on the basis of accumulation of light. For example, less and high. So here the sclera is the outer layer of the eye, and middle is the choroid, and innermost is the retina, which that's contain a rod and cone cell, and may form macula, form fovea. Fovea is the meaning of density of the rod and cone cell, used for the detection of light. And inside is the fluid filled cavity is known as the vitreous humor. So on the other hand, the ciliary bodies contain pars pellicata and pars plana. So here let's begin to understand the microscopic understanding for better understand the here is the pigmented epithelial cell and inner side of the rod and cone cell. So pigmented epithelial cell will filter the light as well as it will transmit the light into the rod and cone cell. Rod and cone cell will detect that light and go into the bipolar cell, and bipolar cell will transmit that signal into the optic nerve. So that is the simple to understand. Why? Let's begin to understand the cone cell and the rod cell. So this is the cone cell, which contains a different segment: outer segment, inner segment, and synaptic terminal. So same structure are present in the rod cell. So the rod cell contains same structure, but the shape will be different. But in the case of cone cell, this cone cell will use for the high light intensity, while the low light intensity, and the dim light, while the high light will detect through a rod cell. So this is the connecting cilium, and this is the rod shape, and in this structure is a free floating disc present inside, and the outer segment, inner segment, that is as well as the synaptic terminal. So you can understand as well. For understanding better, let's draw a bigger picture of the free floating disc. And this free floating disc is the disc contain disc membrane, and I am drawing the disc membrane, so remember about that. So the disc membrane contain a different. So first of all we should understand about the dark time. So not phototransduction or light transduction pathway with absence of light will be rest, and next we will discuss the light pathway during the light time, and the deactivation is the third one. So here is the opsin, and this is the opsin will bind with the 11 cis retinol, that is a type of vitamin A, and to become a rhodopsin. And this is here is the 7 subunit of the opsin contain 11 cis retinol, and here's the carboxy terminal as well as the N terminal. This is the polypeptide, and that's contain N terminal and carboxy terminal, amine and carbon containing carboxy. So here is the 11 cis retinol will bind to become a rhodopsin, but here we will not binding. So here is the rhodopsin formation, but we will not saying that because it is dark. So this will occur in the light reaction when light will fall, and here is the opsin will stimulate to inhibit the GDP containing transducin, which that is contain alpha, beta, and gamma subunit, which that will be inactive. And while here is the phosphodiesterase will be also inactive, and that's function differently. And disc membrane this is. So on the other hand, that is the basically outer membrane contain guanylate cyclase enzyme. This guanylate cyclase enzyme is known as the GC, which that is used for the GTP to convert the cyclic GMP. The cyclic GMP is used for allow to force sodium and calcium to molecule will influx, and this is due to the normal polarization of the cell. While here the potassium and calcium can move out from these potassium and calcium gated channel will remain open. So on the other hand, here is the sodium voltage gated channel will be open in the normal condition in the during the dark time, during without the action potential, without the detection of light. So let's begin to understand the light pathway. So how it's possible. So here is the GDP containing transducin will be activated due to the rhodopsin formation, due to the activated rhodopsin, and when light will be fall on the opsin to become a rhodopsin by binding of the 11 cis retinol, that is a vitamin, and that means it will convert into the GTP active form of the monomeric protein alpha of the transducin will activate the phosphodiesterase enzyme. So the phosphodiesterase enzyme contain alpha, beta, and gamma subunit. So after this binding the alpha subunit with the GTP to basically the GMP, the guanosine monophosphate, and that is basically the guanosine monophosphate will be produced, while the cyclic GMP will convert into the also guanosine monophosphate, which that is come from here. So the also the guanylate cyclase enzyme also present here, which that is produce cyclic GMP also convert by the phosphodiesterase enzyme. Phosphodiesterase enzyme will convert the cyclic GMP to GMP will inhibit the sodium influx from the channel, so the channel will closed. So here is the outside of the cell. So channel will be closed, so the sodium will not move inside. So the potassium and calcium will move outside from the potassium channel also, and to increase the hyperpolarization, the negative charge outside and positive charge inside rather than the positive charge outside. So in this way, here is the neurotransmitter containing vesicle glutamate, which that will be stimulated due to the hyperpolarization of the positive charge inside will trigger the efflux of the glutamate neurotransmitter that will be bind with the neurotransmitter receptor of the optic nerves. After the optic nerve, the synapse will be occur through bipolar cells. So first it will be trigger the bipolar cell, and then it will trigger through a neurotransmitter from the bipolar cell to the optic nerve. So remember about these things. So anyhow, here is the basically the inactivation. So inactivation means the rhodopsin will be the rhodopsin kinase enzyme used to bind with the arrestin by the phosphorylation of the rhodopsin kinase will be phosphorylazed. Due to the triggering of the rhodopsin kinase will be bind with the arrestin phosphorylation, and this arrestin will be deactivate this rhodopsin. After the deactivation, so the alpha and beta subunit and gamma subunit containing diesterase enzyme will be inhibited by the alpha bind with the GTP. After this binding with this, Also there are many protein RGS9, R9AP and GBB5 which that is bind to inhibit this all things, and cyclic GMP will be converted into the GMP will inhibit, and in this way the cyclic GMP concentration again will be again more due to the guanylate cyclase enzyme increase here. So here is the GTP will convert into the cyclic GMP to again influx of the sodium by the positive things. So that is why here is the decreasing of the GMP to increasing of the cyclic GMP to inhibit that things due to the alpha, and alpha GDP will be inactivated. So that is why the GTP will be inactivate to inhibit the cyclic GMP. Inhibit the GMP while the increase the cyclic GMP again due to the guanylate cyclase enzyme activity. While during the activation form, it means the deactivation form, it means the guanylate cyclase will not inhibit due to the inactivation of the phosphodiesterase enzyme, and activation of the rhodopsin, as well as inactivation of the transducin protein. So the phototransduction will be inhibited. So let's begin to understand with the rod outer segment biosynthesis and the retinal pigment epithelium. So this is the rod outer segment we will discuss. What is that? And we will discuss about the vitamin A. And what is the vitamin A? So the vitamin A is the fat soluble which that is come from the different foods, for example carrot, and which the carotenoids, as well as on the other hand the fish oil, and there are a lot of vegetables. So here is the all trans retinal that is present in the rod outer segment, and all trans dehydrogenase enzyme used to produce all trans retinol, because the alcohol hydrogenation, sorry, not dehydrogenation, retinal hydrogenation to become a hydrogen group will be added in this hydrogen ion will be added to form all trans retinol, because the all mean the octahydroxyl group are present. While here is the retinal pigment epithelium biosynthesis of that things. So let's begin to understand. Here is that all trans retinol will move to the retinal pigment epithelium. So anyhow, when we go into the retinal pigment epithelium, it will convert into the all trans retinal ester due to the all trans retinal esterase enzyme. So on the other hand, basically here is you can see this is the formation to become a RPE, that is the RP enzyme 65 is used for the production of 11 cis retinol, because the alcohol group is add, and 11 mean the cis mean that it is that form of the another, it means the bending of the ring chain, hydrocarbon chain. So here is the also the cis retinol also convert 11 cis retinol also convert into the all trans retinal ester. So that things is basically is possible, and this all trans retinal ester will go into the rod outer segment, and this biosynthesis will be occurring in this way. In this way you can see this 11 cis retinylidene, which that is attached with the rhodopsin. After this attached with the rhodopsin is known as that 11 cis retinylidene, and in this way rhodopsin will be bound due to the all trans retinal ester, and this ester will give the basically the nitrogen. Here is the binding with the rhodopsin side, so it is known as the 11 cis. So this is the 11 cis retinylidene, and in this way the rhodopsin will be bound, and in this way here is the all trans retinylidene, which that is bound with the rhodopsin. In this way here you can see, and in the presence of water it can be converted into the all trans retinal. So in this way the retinol is very important, all trans retinol, and that is used for the rhodopsin attachment. So that is why the 11 cis retinylidene is the rhodopsin formation. So let's begin to understand the biosynthesis Walls visual cycle. This is the rhodopsin basically bind with the cis-retinal opsin and cis-retinal as well as opsin. And in this way the cis-retinol or retinal bind with the opsin to give the nerve impulse, while after the dark time dissociation to produce the opsin, and this opsin will also convert again the rhodopsin. So here is the all-trans retinal, which that is alcohol dehydrogenase enzyme used in the liver. When going to the liver, this vitamin A, all-trans retinol formation will be occurs because alcohol. After this is the NADH used to produce NAD plus and all-trans retinol formation. An isomerase enzyme used in the liver for production of again 11-cis retinol, which that is the active form of vitamin A used for the light absorption through rhodopsin. So here is the NADH is NAD plus will reduce into the NADH, and the reductase enzyme used to produce the 11-cis retinal, and that's it is retinal isomerase enzyme used to halt trans retinal and cis retinal. So this is the cycle of the Walls visual cycle. So let's begin to understand the vitamin A. The vitamin A is that there are three forms of vitamin A, which is important for human health. They are retinol, retinal, that is the key player, and the retinoic acid, as well as beta carotene, carotenoids or pigment, and it is present in yellow orange food, is a potent provitamin A. So that is why it's all things is about the function of the vitamin A as well as the light transduction pathway. So I hope you make sense about the light transduction pathway and biosynthesis as a Walls visual cycle. So Walls visual cycle. Thanks for watching.